let me quickly tell about how the antigen and antibody reacts in our body. So in order to look at these, let me start with the slides on the how the pathogen get exposed to the body of the host. This graph explains and relates the how our immune system responds to the infection. Look at this structure. If you look at this, is the graph shows that amount of antibody produced at a different time. What it shows is, if you look at on the top here, there's a first exposure to antigen and second exposure to antigen. So when there is an exposure, there won't be any foreign particle, right? Sorry, there won't be any antibody because you have never been exposed, so never being antibodies are produced. Once the foreign particles or the antigens attacks the host body will produce slowly the antigen. That's why you will see IgM is rapidly rising and then rapidly going down. You will also see that IgG is slowly rising and slowly it is declining. So therefore today, to test any infection is always they look for IgM, because if the infection is very high, it is rapidly it shows the peak of IgM production. So our body produces two different antibodies, IgM and IgG. Now in case in future same virus infects after 2-3 years, our body will understand this pathogen has initially infected. So the memory cells or the B cells, the B cell or the memory cells will remember these and produce the antibody. Look at this time, the antibody productions would be, IgM would be slow and declination will become very fast, okay? But IgG would be produced in plenty and it will keep on producing for a while. So therefore the second immune response would be much faster and quicker and very effective. If it's a primary, then it is slowly produce antibody and their immune response would be quite slow. So this is what you have to understand. If foreign particles get into our body, the first exposure, what happens in the first exposure and what happens in the second exposure. Okay, so now when we talk about the antigen antibody, this is, you can very well equate like lock and key property, where you will see that key rightly fits into the lock to open or to close. Similarly, the antigen antibody binding takes place in that manner. You will see that antigens and antibodies are rightly fit to each other, okay? And now our body normally produce antibody specifically against the antigen, and these production of antibody is called humoral mediated response. If our body produce antibody and act against the pathogens, and that is called humoral or antibody mediated immune response, okay? Likewise, now you know A always have a specific AV. AG means antigen, AB means antibody. So specific antigens or the antibody reacts to each other in a very specific manner. So there are 3 stages where antigen and antibody reacts. First stage is formation of AG to AB complex, so it is called immunocomplex. Second stage is this is very visible, as such they will precipitate or glutinate or neutralize. So that is a second stage where you will see that this substance undergo a reaction. Third stage, you will see the AG and its counter antibody. Two of them would get totally destroyed. All right. So this is how it reacts. Now you see, this is what the complex is. The first complex form is AG-AB complex. Okay, and here what you have to understand is, once the complex is formed, then it triggers further for reactions, and this happens when there is an inflammations or cytotoxicity. You will see that inflammations, at that time this complex is being formed and will get destroyed. All right. Now yeah, this is just for further explaining that how AG and AB becomes very specific in their reaction. You also know there is an epitope and paratope part of the antigens antibody, and this in fact helps them to bind each other. In case of IgM, you know there are 10 different sites where these epitope can come and combine. Likewise, now the third, yeah, this is just a process of explaining how these AG and AV binds to each other. They need a certain force, and this force is called hydrogen bond, hydrophobic bonds, and van der Waals force. These are the force which keeps them together. Okay, and once this complex has been formed, these bonds slowly breaks off, and that's how it gets destroyed. Now avidity is referred to overall capacity of an antibody to combine with multivalent antigens. Here, when when the antigens has got many sites for binding antibody, then many antibodies binds on a same antigen at times. This is not all time, this is at time. So this is called multivalent ability of an antigens, where it can combine with many antibodies. Otherwise, normally the antigens will have a specific antibody. Next is, now we will look at different types of antigen and antibody reaction. This is quite easy. The first one is the precipitation. Precipitation is when you add antibody over the antigens, you will see there is a clogging. It forms a clumping, and ultimately this clump will initiate further destruction. Okay, now this is how it looks. Say antigen and antibody join together and it has clumped together. So clumping would happen initially. There will be lesser clumping, and you will see the highest, and it gets finally distort, or if you further add it, there will be no more clumping. Likewise, second reaction is agglutinations, where you will see a sudden antigens will try to go around the antibody, and then it will not allow. And when these antibodies are surrounded by many antibodies, that is how you are going to see is becoming clustered, or the clump of antibodies. Likewise, this is how it happens, the clumping. You just look at, this is free antigen antibody. This is all together, joined together by a antibody or antigens. Third one is cytolysis. Cytolysis is a breakdown when the complex is being formed and is being activated. These antigen and antibody complex will get slowly destroyed, and this is called cytolysis. Last one is an opsonization, when the foreign particle is combined with the antibody. These complex becomes very vulnerable to attack by the another antibody or the other phagocytic cells. So if foreign particles or the antigens is being combined by the antibody, they become very vulnerable to the breakdown, and during that time these all the phagocytic cells comes and combines with this complex and further breaks down, and this is called opsonization. So antigens become more susceptible to the phagocytosis. Phagocytosis is called opsonization. With this, I hope this has helped you to understand about uh, different types of antigen antibody reaction. Thank you.